You know how DNA is structured and how it controls life? In this video, we will understand the complete structure of DNA in a very simple way. Let's start with the basic structure of DNA. DNA is a polymer of deoxyribonucleotides. This means it is made up of repeating units called nucleotides. Each nucleotide contains a nitrogen base, a sugar, and phosphate group. The four types of nucleotides are adenine, guanine, cytosine, and thymine. These nucleotides are connected by 3-5- phosphodiester bonds to form a long chain. Now, according to Chargaff's rule, adenine equals to thymine and guanine equals to cytosine. The DNA double helix was discovered by James Watson and Francis Crick in 1953. It looks like a twisted line. The two strands run in opposite directions. This is called antiparallel nature. One strand runs 5- to 3- and other 3- to 5-. Hydrogen bonds hold the strands together. Adenine pairs with thymine by 2 bonds. Guanine pairs with cytosine by 3 bonds. The DNA helix has a major and minor grooves where proteins can bind. Features of DNA helix. DNA is a right-handed double helix. It has a diameter of 2 nanometers. One complete turn has a 10 base pairs and length of 3.4 nanometers. The outer part is a hydrophilic due to phosphate backbone and inner part is a hydrophobic due to bases. DNA strands are complementary, not identical. The genetic information is stored in one strand called template strand. DNA exists in a different forms: bDNA, aDNA, and zDNA. bDNA is the most common form in the body. zDNA is a left-handed and has a zigzag structure. Some special DNA structures also exist like triple-stranded DNA and four-stranded DNA. These structures are less stable and seen in special conditions. DNA molecules are extremely large. They are measured in a base pairs. In humans, total DNA length is about 2 meters, but it is packed inside a teeny nucleus. Denaturation means separation of DNA strands. It happens due to heat or change in pH. Only hydrogen bonds break, not phosphodiester bonds. The melting temperature is the temperature where 50% DNA becomes a single-stranded. Higher guanine cytosine content means higher stability and higher melting temperature. DNA is tightly packed inside the nucleus. It forms chromatin and then chromosome. In eukaryotes, DNA wraps around the histone proteins to form a nucleosome. It has a biomedical importance. DNA is the storage of genetic information. It controls protein synthesis through RNA. Some drugs act on DNA. Anti-cancer drugs like a 5-fluorouracil inhibit DNA synthesis. Cisplatin damages DNA structure. Purine metabolism produces uric acid which can cause gout. Substances like caffeine and theophylline are pharmacologically important purines. If you found this video helpful, like, share and subscribe to Recall Right Official for more pharmacy concepts made easy. Thank you so much.